First, I would like to thank the organizer for giving us the opportunity to talk about a new resin, Nuvia IMAC, and also how we use this for the purification of bacterial proteins. And we try to build a purification platform. As we know that for MAPS, we have the protein A-based platform for decades, and we pretty much enjoy using the platform for most of the molecules. But so far, there's no platform available for recombinant proteins. And we are trying to see if we can come up with something similar as efficient as the protein A-based platforms for recombinant proteins. So many of us, when we were in graduate school, we more or less used IMAC for purification, because at that time, we only need a small amount for activity tests for crystal structure. In this week, we come here at BBI, we are talking about mass production of drugs, protein drugs. So I think I probably don't need to talk too much about IMAC, as you all know about this, so the overview will be really brief. But I want to mention the additional criteria for chromatography resins used in process chromatography. Because we are talking about large column, we need to process huge amount of material. So the beads must have some special features in order to meet all those requirements for production. And then I'm going to talk about a case study when we use Nuvia IMAC for the capture of the target molecule and how we deal with the impurities unique to this process. And at the end, I will wrap up my talk and with some thoughts about this new platform. So here is just a diagram showing you the basic idea about metal affinity chromatography. We immobilize the ligand on solid base beads, and after that we charge the ligand with metal. You can, the most popular one is nickel, and we also see the use of other metals. Depends on the target molecules you're trying to purify. And when the target comes in, either they could be natively rich in histidine or cysteines, or for some of the phosphorylated proteins, we also have seen that the phosphorylated group interacts with the center metal, and we can pull out the target molecule from a mixture. Yeah, I also seen affinity purification for calcium metals, proteins with affinity for calcium, for zinc. So it depends on the sequence of your molecule. You can purify a lot of proteins using the same principle. But the most popular way is to engineer a histag as a fusion protein with the target. But no matter what proteins you're trying to purify, since we are dealing with affinity, so like protein A, you probably don't have to do a lot of feed manipulation. You just load the sample at close to neutral pH, wash out the impurities, and then elute the target molecule from the resin. So although we use this a lot in the lab, but in fact in manufacturing, we have cases like factor A has recombinant factor A, one of the purification steps, affinity chromatography was used. And also we have seen some single-train variable fragment-based constructs, in which the IMAC was used as the first step for the capturing. And also lately we saw cases for example, recombinant human interleukin-1 beta was purified using IMAC. The latest report I saw is 50-liter column for the capturing of this molecule. And also some of the vaccines in clinical trials are purified by using IMAC affinity chromatography. And as I mentioned earlier, you can purify native proteins if they're rich in histidine or cysteines or those carboxylates. You can also make the histag fusion proteins, and the expression host can be E. coli, yeast, or insect cells, depending on the folding of the protein and the expression level. Now, let's talk about process resins. Because we need to process a lot of material, that means we have to run quickly under dynamic conditions. So the beads have to be strong when you pack a large column. And for quality control reason and for reduction of production cost, we want to reuse the column as many times as possible. So that means we need to be able to regenerate the column, and after the regeneration, you still are talking about the same binding capacity, selectivity, and target recovery. And a lot of time, we also, for IMAC, we need to use other kind of chemicals in the buffer in order to keep the protein active, folded correctly. And sometimes we also attempt to do refolding of the target molecule on the resin before we do elution. That means the IMAC resin also has to be compatible with a lot of chemicals like reducing agents, denaturants, detergents. So we'll see how I'll show you data our Nuvia IMAC is stable or compatible under these conditions, and it survives all the stringent CIP, SIP conditions. And the resin has to be able to pack into a large column easily, will not collapse, will not generate high pressure, and you can run the production at high flow rate without seeing a column get packed and the flow stops. With this in mind, I just want to show you the dynamic binding capacity of this resin at different flow rate. So here is the Nuvia IMAC curve, and we try to change the flow rate for the loading from 100 centimeter per hour up to 300 centimeter per hour. So for the configuration of the test protein in the column, at 300 centimeter per hour, we are talking about just 1 minute of residence time. And you see here that binding capacity is not compromised much, and pretty much is remain at 40 to 50 milligram per mil binding capacity. So we optimize the pore size and the pore structure to allow the target molecule penetrate into the beads, even at high loading flow rate. And now talking about regeneration. So for process, of course, in order to reduce the costs, we want to run the same column after it's packed as many times as possible. So in this study, we try to mimic a real-world purification that you have accrued from, for this case, it's from E. coli. So we try to set up a cycle in which we load a sample after the purification. We use EDTA to strip the nickel and then apply sodium hydroxide to clean the column, and then recharge, and then load the next round of E. coli lysate. And then we also park the column in sodium hydroxide for a while, and then load another batch of E. coli just to see with different lots of feed material whether we're seeing consistent binding capacity. And now you see here, it's from cycle 1, we just went all the way to 102. Since we didn't see much difference in binding capacity, we just stopped. It's just like an energy bunny, keep going on and on. And you see there's not much, besides the variation of the feed, we don't really see any change of the dynamic binding capacity. Now we come to the purity. So we are comparing the purity of the finished purified material of the first cycle and the cycle 102. As you can see, the purity on the gel, there's not much difference from the first cycle to the last cycle, and we are also talking about the same yield. That's because we actually engineer the surface of the beads. We're trying to keep it as hydrophobic as possible, but no charge. So the impurity proteins are not bound through non-specific interaction with the polymer backbone. On the other hand, the target molecule is also only bound through the chelation, not by charge interaction or hydrophobic interaction. That means when you hit the resin with imidazole, you are releasing those molecules bound through chelation. Mostly it's the target protein. And that can also explain for this data. It's a lot of time when we're trying to process a lot of material, we want to run it quick. If the mass transfer is not efficient enough, some of the target molecule will be excluded out of the beads, and we lost the material in the flow through. So that's one of the reasons that we don't always get good recovery of the target, just because during the loading you have leaching of the target in the flow through because of the poor mass transfer. For the IMAC case, we just try different formulation to see where we hit the best surface area that we have good binding capacity. At the same time, we have good pore size to allow the binding really happen when you run it at fast flow rate without much leaching in during the phase when you start loading until you hit the 10% breakthrough point, as a way to improve the recovery of the target. So another contribution to the target recovery is the elution. Because the elution, as I mentioned earlier, if some of the target molecules are bound through non-specific interaction, when you use imidazole or histidine, in some cases for the elution, you may lose some of the target due to the non-specific interaction. In this case, as you can see, like the recovery is like close to 97%. That means we also like achieve a surface property that we don't really have non-specific interaction due to the polymer's backbone, so as a way to improve target recovery. Now these are the some of the chemicals we've tested to be used for the IMAC purification. So basically you can use most of the common reducing agents you may need for keeping the protein functional. And I also list some of the detergents, of the ones we often use in products production, not that nasty. There are cases showing like additional wash with this detergent is helpful for endotoxin removals, because some of the endotoxins are so hydrophobic, they just interact with the proteins to be purified. In case you need to do refolding, and we found that the tolerance of vanadine hydrochloride to 6 molar and then urea to 8 molar without compromising the binding and the selectivity of the resin. And then if you want to use non-phosphate buffers, you also have the choice of the good buffers in the range of pH 7 to 8 without compromising the binding capacity and also without causing too much nickel leaching from the resin. And of course, a stable in sodium hydroxide. We do have a complete list of chemicals that can be used together with IMAC and our brochure. If you want more information, we can talk after this. Now about column packing, because we are so familiar with gravity flow column, we just pour the resin, drip the sample through the column, and then apply the elution buffer, we get the target. But for process, the quality of the packing actually affect the quality of our final product purity and also the consistency from lot to lot. And you also want to pack the column and then run until you finish the campaign rather than having to deal with the repacking if it shrinks or it cracks during the production. So we are trying to see, like from a small scale, you have just 1 mil column, all the way to like a 6 to 7 liters, 20 to 20 column, how the resin is packed. And we use like columns from different vendors, like from GE, from Minute-Pour, and also our own column in place to see how that affects the packing using a flow pack or axial compression. As you can see, like no matter which column, what size, you can easily pack the column with good performance. In additional study of the packings, like you might have to deal with, depends on the column's configuration, you may be using different percentage of the resin slurry, and we found there's not much difference, like from 45 to 65%. And also you can pack, when you do axial compression or flow pack, you can run the packing at different flow rate without compromise on the column's packing performance. And here is the pressure flow curve for a 20 by 20 column. Because like when we are developing resins, a lot of time we have a hard, like troubles trying to balance the binding capacity versus the flow properties. Because when you have a smaller beads, you have a larger surface area, you can have larger binding capacity. On the other hand, when we work with small beads, we a lot of time run into pressure problem. You just cannot run fast. That actually, the benefit you get from a higher binding capacity is not going to be harnessed if you have to slow down the load in order to have the protein bound, or that's going to affect the productivity. So we try to see, like, we actually control the size of the average bead particle size at around 50 micron with a very narrow cut. So that helps reduce the column pressure. When we pack a large column and run at 300 centimeters per hour, we still keep the column pressure at well below 3 bars. It's around 2 bars or slightly below 2 bars at 300 centimeters per hour. So after I finish all those basics of this IMAC resin designed for the process purification, I want to spend some time on the purification of a bacterial protein from E. coli crude extract. Because as we all know, that the metal is not covalently immobilized on the resin. That means part of it will be leached out during the purification, depends on your buffer composition. And how are we going to deal with this? As we all know, we have allergic reaction to nickels. And also for the E. coli protein, if we purify protein from E. coli, a lot of time we're concerned with the endotoxin contamination, because it's for sure, E. coli has a lot of this. So I want to focus on these two impurities because we are familiar with removal of HCP whole cell DNAs already. So here is the IMAC used for the capture of the target molecule from E. coli lysate. The first lane 1 shows you the expression level and the impurity level of the crude material. And then as you see my buffer composition, basically you just load the crude on the column pre-equilibrated with the binding buffer. You wash with the same buffer, and then you hit it with high concentration of imidazole, and we're done. And as you can see on the gel, it's quite pure already. So a lot of time when we do recombinant proteins with our affinity approach, we have to run many columns in order to achieve this kind of purity level. On the other hand, there are impurities that are not shown by a gel. The nickel I just mentioned is one of them, and then the endotoxin and the DNAs. So for purification of proteins from E. coli expression host, we do need to do polishing. So for this purification, we actually screen through the resins we have in-house. We looked at the anion exchangers, we looked at the cation exchangers, and we also have the CHT, that's a metal that affinity plus cation exchange. It's a mixed mode resin. It's interesting, we try to compare, although Nuvia Q and MicroPrep Q, they are all strong anion exchanger, but we know from our past experience that the MicroPrep High Q has some sort of hydrophobic interaction from the backbone. And we have seen reports that this actually helps the removal of endotoxin. So in our case, we want to see if this is true. And then for the cation exchange, we also compare a purely strong cation exchange versus a hydrophobic cation exchanger, Nuvia C' with an organic ligand to provide the negative charge to see how well they are for separation of other process-related impurities. So as we know, the PI of the protein is at 6. So for the Q resins, the target is bound and eluted when we are ready, finish the washing. And then for the cation exchangers, as we expect, the target was in the flow through. So for CHT, it's bound and eluded. Then we compare, because we want to focus on these two types of impurity removal. So as you can see for the endotoxin, in fact, we did see a better, maybe 1 order, or slightly more than that, because the dilution was not exactly perfect. So we saw like the macroprep high Q gave better endotoxin clearance. And then for the Nuvia C prime versus Nuvia S, similar endotoxin clearance. On the other hand, we have data not shown here as the Nuvia C prime has better HCP clearance efficiency. And also we like the fact that it removes nickel better than the Q resins, as we all understand it. So with this information, we built a purification platform in which we use Nuvia IMAC for the first step, the capture step, and then use the Nuvia C prime for intermediate polishing, and then the macroprep high Q or CHT as the final polishing. So as you can see here, the reason why we lay out this way is like we want to use a cation exchanger to take care of the basic species, and then use the macroprep Q or CHT for other acidic impurities. Plus, if you look at the buffer compositions, from IMAC elution, we can actually load this directly onto the Nuvia C prime because the target is in the flow through. And at this low ionic strength, a lot of the whole cell proteins, the basic ones, are bound by Nuvia C prime and removed. And in the last step, now this time we have the target bound. We further remove acidic species, and there's no need of buffer exchange. So quickly you can chain all this column together and finish the run after you set up our chromatography instrument. So this is the initial results for the workflow comparison. You see that the workflow with IMAC, Nuvia C prime, then CHT1 actually gave better clearance of impurity compared to the macroprep high Q as the final polishing. But what's even more interesting is like as I mentioned earlier, that we saw the Nuvia C prime remove metals. And as we know that the Nuvia IMAC elution simply rely on the imidazole. So we can have the target bound on the column and adjust the buffer conductivity. So when we do the elution, we can have a low buffer conductivity to do flow through on C prime. We want to see if this actually affect further, like reduce the metal content in the C prime flow through fraction. Because you see the structure of the C prime's ligand, it has a carboxylate. Basically, we are presenting a lot of carboxylates on the resin as a metal scavenger. And this is the data. We found that if we further remove that 25 millimolar of sodium chloride in our equilibration buffer for C prime, we actually can further reduce the nickel content 10 times. Now this is a better workflow now. It's like we have two washes. The first wash on the IMAC, we use a still buffer with salt to remove the impurity proteins. And after that, we do another wash to prep the material in a low salt environment, and then elute the target using imidazole with a buffer of low conductivity. And now the C prime is done in tris instead of phosphate, and then this material can be loaded directly onto the CHT1, and we elute the target at high salt. So after this, we actually submit a sample for ICP analysis. There is no detectable amount of nickel. And then the endotoxin level, so the nickel content is okay. So the more traditional way for removing nickel, of course, is to do diafiltration. Because according to my conversation with a customer in the past, when they use IMAC, when they're trying to reduce the nickel content using diafiltration, they actually, for one part of your feed, you have to do over 22,000 liters of buffer in order to effectively reduce the nickel content. So that means a lot of water consumption. But with this, you pretty much do the straightforward purification without any buffer manipulation as a way to reduce the production cost. So So the data here shows that with the reduction of the equilibration buffer for C prime, we can facilitate more efficient nickel clearance in the purification. And then for the endotoxin removal, I would like to put a question mark here. So if we assume this is a molecule to be used as a vaccine, with this kind of endotoxin level for a 50 microgram dose of vaccine, it's way below FDA's limit, 300, I believe it's 350 EU per 50 microgram dose for a 70 kilo human body. But if we are using this to purify injectable proteins, then I would like to further improve the endotoxin clearance efficiency. So now I want to conclude my talk. As I demonstrate, we can use the Nuvia IMAC for the capture of target molecules. It offers the purity, the recovery, and the robustness for process purification of proteins. And also, with this case, we also show you how to deal with the process-related impurities from this kind of purification, mainly as the metal leaching from the resin, and also there might be contamination from the expression host. You may not have this problem if you use CHO cell to do expression, or you express the target in insect cells or yeast cells. But anyway, with this platform, we can take care of this kind of process-related impurities. So when I was in graduate school, I never really thought that IMAC could go to large scale because of the concerns about the nickel, and also the need for some applications you need to remove the his tag. But as we move on, these days I have seen reports like there are GMP grade enzymes for the removal of the his tag, and I think QIAGEN has demonstrated many cases that they can remove the tag and they can remove the protease using a second round of IMAC. Or you can do the digestion in solution before you really do the purification to remove both the enzyme and the his tag after the digestion. So also, if we just simply look at the price quote, we feel like the IMAC is more expensive than most of the resins except protein A. On the other hand, if we look at it from a different angle, like with a IMAC resin, you pretty much cut several steps of purification into one step. So that's a lot of saving in terms of production cost. Now we are talking about expanding our drug molecules from MAPs to other recombinant proteins. So we do see the need that we need to develop a purification platform like the protein A-based platform. We have the need, and now we certainly see the possibility that we have the resin. Once we have a cheap, efficient enzyme, we should be able to deal with the tag removal and get the same quality of drug molecules for more patients. I would like to conclude my talk and want to thank my colleagues in Bio-Rad. And also, if you need more information about the resin, you can go to our website to look for the protein purification solutions. And I'm open for questions if you have any.